Ladies and gentlemen, we're on the home straight now, and I'm going to ask Marita Avery to introduce the David Marsden Prize and indeed the David Marsden Award winner. Thank you, Marita.
David Marsden Award 2019. Maybe some of you in this room met Professor David Marsden. Unfortunately, I did not. He was the Dean of the Institute of Neurology in Queen's Square in London. And he was one of the prominent founding members of the Movement Disorder Society. And many movement disorder experts across Europe and beyond first learned about dystonia from Professor Marsden. He especially encouraged young scientists to become involved in the field. When Dystonia Europe was founded in 1993, Professor David Marsden gave us a lot of good advice and took a genuine interest in the development of our organization. It was therefore entirely appropriate that David Marsden should be honored by an award given by patients in his name. We are assured that he would be very pleased to be associated with the encouragement of young scientists in this way. The world of neurology, and especially dystonia patients, are grateful to him, and we hope this award will inspire many more young scientists to do dystonia research. We thank Ipsen for their generous support of this year's award. It is an honor to present the 9th David Marsden Award to Dr. Anne Weissbach from Institute of Neurogenetics in Lübeck, Germany, for her paper, Alcohol Improves Cerebellar Learning Deficit in Myoclonus Dystonia, a clinical and electrophysiological investigation. Please join me here, Dr. Anne Weissbach. I want to sincerely thank you for the paper and your dedication to this study. And with that, I give you this award of 10,000 euros. Wow. Wow. Okay. Thank you. So now I give the word to Anne that will talk a little bit more about this study.
Wow, hello everyone. I'm unbelievably honored, and I can't tell you how thankful I am to receive this award and to have the chance to speak in front of all of you. I'm Anne. I'm a clinician-scientist and an adult neurologist from Lübeck, which is at the north of Germany. And I'm very, very happy to present to you today the study results of our study that we did in myoclonus dystonia. And myoclonus dystonia in a lot of patients is caused because of mutations in the so-called epsilon sarcoglycan gene. You see it here as SGCE, that's the abbreviation for the gene. And this gene is important for a protein which is in the membrane. So it's important for the stability of the cell and also for cell trafficking. And what other researchers found out before was that SGCE is actually expressed in several parts of our brain, and especially in the cerebellum and in the neurons of the cerebellum, especially in the so-called synapses. And these are important structures of every cell to communicate with other cells. And what is also interesting is that when patients inherit these mutation, and they inherit it from their mother, that the gene mutation is silenced. So they are most likely not express the motor symptoms, but of course they have a high chance to pass the mutation on to their children. And if you have such a mutation and you have the motor symptoms, you most likely show symptoms in really early childhood. And these are most likely myoclonic jerks. And I will show you how these jerks look with the help of one of our patients that participated in the study and that we know since a really, really long time. And these myoclonic jerks in the majority of patients are combined by segmental or focal dystonia, mostly cervical dystonia or writer's cramp. And also, as we heard before from our experts around here, also this kind of dystonia shows psychiatric symptoms. And what is also interesting for them is that mutation carriers that even if they don't show the motor symptoms like myoclonus or dystonia, they also can have psychiatric symptoms. What a lot of patients told us, and what was really interesting to us, was that they have an alcohol responsiveness. So when they drink alcohol, their symptoms improve. And this was really interesting because a lot of the oral pharmaceutical treatment options that we have, and I just put some of them here, do not have the same good effect as our patients know from alcohol. Another important treatment strategy in these patients, although, and we also heard from our experts about this, is deep brain stimulation. For example, of the globus pallidus internus, you see this here as GPI, or VIM, the thalamus, parts of the thalamus, and they show quite good results. So in previous studies in humans, in patients, we saw that most likely subcortical structures of our brain, for example, the brainstem or the cerebellum, are likely to be a cause of the disease, so causing the symptoms. And most likely, and you also heard this before, and I'm very thankful for this, is that probably plasticity, so the way the brain learns, is kind of abnormal in patients with dystonia, and especially with myoclonus dystonia. So we were actually quite interested in the plasticity of structures like the cerebellum and the brainstem, or the brainstem and the basal ganglia. So we wanted to know how communicate these parts of the brain with each other, and how maybe this communication and these learning of these brain structures maybe is different in patients with myoclonus dystonia that have these mutations in the SGCE gene that is expressed in, for example, in the Purkinje cells of the cerebellum. So we were looking for nice ways to examine our patients not only clinically but also neurophysiologically. And we knew that the blink reflex is actually transmitted in these parts of the brain. And I'll show you on that slide how actually blink reflex conditioning, which is kind of a measurement for cerebellar motor learning, works. Probably all of you experienced that when you get an air puff to your eye that you blink reflectively. And if you combine these so-called unconditioned stimulus of the air with a conditioning stimulus like a tone, like a beeping tone, and you do that several times, you get conditioned, so you learn. And then you can suddenly blink, although you don't get the air puff, but you only get the tone. And this so-called motor cerebellar learning is due to a certain way of plasticity. Till now, we are not completely sure where in the brain this plasticity takes place, but most likely it's in the Purkinje cells, as I mentioned before, the neurons of the cerebellum, or the deep nuclei of the cerebellum that are kind of the output of the cerebellum towards other brain structures. So we used these paradigm in our study and combined it with a clinical examination of our patients, because we wanted to correlate our clinical as well as our neurophysiological findings. And clinical examination means that we took a video of all of our patients and we showed these videos to movement disorder specialists from all over the world. And we did this before and after alcohol application, because we know that alcohol is kind of a GABAergic agent, and GABA is a transmitter, so kind of a hormone in the brain, which is important to communication in the brain. And we did this in our myoclonus dystonia patients, but also in age and sex-matched healthy controlled subjects. And at the first day, we just did our baseline measurements. So all the measurements I explained to you before without the patients receiving alcohol. And then we repeated all the measurements in exactly the same way after the patients receive alcohol. And when I say they receive alcohol, they receive that in a standardized, safe way. So we gave ethanol to the patients, and we calculated the amount of alcohol to cause a 0.8 per ml breath alcohol concentration with the help of a so-called Widmark formula, which encounters the sex and the weight of the patient. And we mixed that ethanol actually in a soda, which was decaf and sugar-free. And this was a protocol that was also established before in patients having a tremorus, a so-called essential tremor disorder, and this is also responsive to alcohol. And I would like to show you with the help of one of our patients here how the disease actually looks like. So this is one of our young patients. She was 20 years old, and you can see that she has severe myoclonic jerks. They're generalized, but they're mostly in the upper part of the body, affecting the trunk, the arms. She also has cervical dystonia, causing a retrocollis, and in a few seconds you will see that this is also severely affecting her motor function, like when she wants to eat, when she wants to write. She also has problems writing, but not only due to the myoclonic jerks, also due to writer's cramp. And on the next video, you can see her. Let me shortly go back. Let's get it started. On the next video on the left, yeah, it's working. You can see that her myoclonic jerks are much better. So she's not jerking as much in the whole body. Also, the dystonia improved a little bit. But what you, I think, realize is that her symptoms were not gone completely. And this is what most of our patients during the study told us, that if they would consume more alcohol, they would probably have less symptoms. But we stick to this standardized way, and we wanted to achieve the same concentration of alcohol in all our patients. So I told you that with the help of international movement disorder specialists that were blinded to the status of the patient, we were measuring how the motor symptoms improved, and we used standardized rating scales for dystonia and for myoclonus. And we found out that the myoclonus, and you can see that on the screen, that with a C2, like off C2 means without alcohol, on C2 is the patient having the alcohol, that all symptoms significantly improved, but the symptoms that were, all symptoms improved, but the only symptoms that improved significantly were the myoclonus symptoms. And we were rating this with the so-called UMRS, the Unified Myoclonus Rating Scale. And they improved for the total amount of score of the Unified Myoclonus Rating Scale, but also for the subscales, so for the action myoclonus, when a patient is doing something, and especially during the functional test that patients do, like pouring water in a glass or bringing a spoon to the mouth. And the dystonia rating scales, here like the Berg von Marsden rating scale, the Toronto Western Spasmodic Torticollis rating scale, and the writer's cramp, improved, but not significantly. So, what did we do during our neurophysiological examination? I told you that we were interested in the blink reflex conditioning. And here you can see me wearing our setup, actually. And I'm wearing here the air nozzle, which applies the air puff. And I'm wearing the headphones that are giving the tone. And you can see here that when I get the air, that I blink. This is a surface EMG, and it produces a nice blink. And when I'm actually conditioned, so when I receive the motor learning paradigm for at least several times, so we had 10 acquisition blocks, which means in these 10 acquisition blocks, in each block 10 times this tone was combined, then I'm actually blinking when I only hear the tone. You can see here the tone starts and I'm blinking, and this blink appears before I get the air. So then we were analyzing all our data and comparing it, not only for the 2 days, but also between groups. And what you can see actually is that our myoclonus dystonia patients, you can see them here in the black circle, get conditioned so they learn significantly lower than our healthy controlled subjects. This is the percentage of conditioning, and this is over the 10 blocks. So during the time, all increase, but the myoclonus dystonia patients are much worse. And this is after alcohol. And interestingly, our healthy subjects got worse under alcohol. If they wouldn't receive the alcohol, they would get much better. So we also had healthy controls that didn't receive alcohol, and they were much better after the repetition of learning, which makes sense, right? But interestingly, when our myoclonus dystonia patients got the alcohol on the second day and learned a paradigm again, they were improving under alcohol. So the opposite compared to the patients was, compared to the healthy controls, was true to them. So what I wanted to show you was that not only that the blink conditioning was abnormally reduced in our patient, it improved after alcohol intake. And this was also true for the symptom severity of myoclonus, that significantly decreased after the patient received the alcohol. And the neurophysiological data and the clinical data was correlated in a certain way. So patients that have severe action myoclonus actually learned much worse than others. And we actually suspected that due to their mutation that the patient carried, the so-called GABAergic deficit in the neurons of the cerebellum, the Purkinje cells, was present, was there, and maybe prohibited them from really learning in a normal way or exploring the plasticity of this part of the brain in a normal way. And maybe through this short time substitution of a GABAergic agent, of a GABAergic agonist, we were able to shortly ameliorate this deficit. So for us, actually, SGCE positive myoclonus dystonia seems to be a kind of a model disease. So we heard from our speakers before, and all of you know that dystonia is really heterogeneous, and our monogenic forms of dystonia, like these SGCE positive myoclonus dystonia, can be a really nice model because all the patients have the same mutation, although they probably exhibit different kind of severity factors, different kind of motor symptoms. The cause of the disease is the same in all of them. And I showed you that we were, with this paradigm that we did here, we were actually investigating subcortical structures, so the interaction of subcortical players. And we actually don't know much about the pathways from the cerebellum towards the primary motor cortex. So this is actually also still an open question. And this is what we would like to follow up in our upcoming study. And this is where also the David Marsden Award will really help us. So we are planning to continue our research in a multimodal way with the myoclonus dystonia patients, with having, again, a clinical examination, but also bringing objectified measurements like sensors within these measurements. And then we will use also imaging to show a potential deficit, and transcranial magnetic stimulation that I also did over the last years to investigate the excitability and the interaction of the different motor network players. And with the help of another learning paradigm, so-called motor sequence learning, we are able to investigate cerebellar and cortical, primary motor cortex and premotor structures. And this time we will not use alcohol as a changer of plasticity, let's say. We would like to use non-invasive techniques to induce this plasticity. So just really briefly, here you can see a picture of a study which uses these sensors to measure the severity of dystonia, for example, when reaching out and grasping a certain object. And here you can see the transcranial magnetic stimulation in one of my students. And you can see that we use infrared tracking devices. So we really have the possibility to target our stimulation areas under these coils, and here you can see one coil over the cerebellum, one coil over the primary motor cortex, and one coil, a small coil, over the so-called premotor cortex that is also really important in shaping and preparation of movements, as is the cerebellum. And this we will all do in a neuronavigated way. So we will use the individual MRI of each patient and healthy control to define where are we actually stimulating. Because if you see here in the cerebellum, there are certain areas that you can nicely reach from outside with a plasticity induction tool. But there are others, like the lobus 5, that would be quite interesting for us, but it's in the center of the cerebellum. So we are hardly reaching it, and if we would stimulate much other areas before. But also these lobus 8A is quite interesting because it plays an important role in motor learning. So where are we now? These plasticity protocols, and I just want you to see that there are multiple of them, 4 of them. We already tested in healthy controlled subjects. In 30 healthy controlled subjects, we applied them, we tested them of efficacy using transcranial magnetic stimulation, and are right now analyzing our data. And the most promising one we will use in our myoclonus dystonia patients. So I couldn't do this all by myself, and I'm unbelievable thankful for the great people I can work with, especially for my supervisors, Christine Klein and Alexander Münchau, who I know for a really, really long time and always supported me, and also for the team. And special thanks to all the patients that always participate in our research. Without you, this would never been possible. And it's really wonderful that I was allowed to present here to you, to receive this award, to follow up in our upcoming study. Thank you very, very much.
Dr. Weissbach, don't go away just yet. I think we've got time for a couple of questions. While you're all thinking, this is another example of how alcohol has repeatedly come into the professional and academic subjects for study by neurologists. Another area that used to be studied with alcohol is hyperkalemic periodic paralysis. I gather in Newcastle they used to drink crates of Newcastle brown ale to see whether they could induce the hyperkalemia. And obviously your patients must have enjoyed themselves immensely. I've got a question for you. It seems to me that the GABAergic functions tend to be CNS depressing. Mm-hmm. And with the translation from a pharmacological ethanol model to a stimulation model, are you trying to induce long-term depression or long-term stimulation of the Purkinje cells in the cerebellum? What is the if you like, the biological, physiological thing that you're attempting to do with that stimulation?
Yeah. So you mean with the stimulation of giving alcohol to the patients, or with the new plasticity protocols that we
Well, the new plasticity protocol using, transcranial magnetic
So first of all, thank you very much for this really important question. Actually, it depends on the plasticity protocol that you use. But actually, the general behind that is that we expect the Purkinje cells to work abnormally high. So they kind of really inhibit these, I told you before, these deep nucleus of the cerebellum that have the output. So they inhibit them, so they can't function well, and they actually involve over the dentato-thalamocortical drive. They involve actually and influence our motor areas of our cortex. So what we are hoping is, if we tune down Purkinje cells and in their output, that we can really kind of have an effect on other motor players. That's actually the goal. But actually, this question that you ask me, I cannot answer that yet. And this is why we actually have transcranial magnetic stimulation as a tool not only to induce plasticity, but also to see what this plasticity is doing. So we will use dual pulse and single pulse TMS to measure the excitability of the primary motor cortex and also the cerebellar cortical interaction. So we will have our output players, and we can nicely then later see not only via the clinical data, but also via the TMS and via the imaging data, what kind of effects these plasticity protocols have.
Yes. I can see the alcohol industry are going to close down your program very quickly because you get all the benefits of the alcohol without all the dangers of liver disease and all the other things through the neurostimulation. Has anybody got any questions for Dr. Wiese? Yes, please. Just have a quick.
Yeah, so I'm having writer's cramp and I'm having cervical dystonia. And I agree, I take alcohol content, I take alcohol and it's positive to me, but it's not always positive to me, the alcohol. So particularly what content should I take from the, and which one should I take? In the sense, if there is any suggestion as per your research, currently, there is no medicine available.
So this young man says that he takes alcohol for his writer's cramp. But he's not sure what sort of alcohol he should be taking.
So what I would really like to point out, and this aspect is really important to me, is we didn't use alcohol in this study as a treatment.
No.
We used alcohol to influence the excitability of the cerebellum. So to change probably the transmission status of the cerebellum and within the Purkinje cells, just short time. Because we already knew that from, for example, people who are addicted to alcohol or children who have suffered embryonic alcohol toxicity, that they are quite influenced via their plasticity that is examined due to the eye blink conditioning. So actually this is why we decided to do this one-time alcohol treatment. And you know, the majority of patients always reports to us that alcohol influences their symptoms. And we also wanted to know, first of all, how, so to see clinically how this affects, and maybe to find out and to also tell them an answer how alcohol kind of influences their cerebellum and subcortical structures and how this maybe is benefiting to the symptoms. But of course, this is not meant as a long time or not even short time treatment option due to the severe side effects.
So this research is only applicable to the muted gene respective dystonia right now, what you're doing? So you pointed to the particular genes, right? Particular DY-T1 gene dystonia, right? Or is it to all dystonia, all category of dystonia, or is it only for the genetic dystonia?
So sorry for not making this clear. So these were patients that had an SGCE mutation. So they were monogenic dystonia forms. And we would like to see them as a kind of model for other, so far not yet well-defined genetic disorders. Yeah.
Thank you.
Thank you very much indeed, Dr. Weissbach. I'm sure that you'll be coming back to this meeting giving us your latest results.
Definitely, that would be a great honor if I
having
could get
entered
a chance.
the field of one of the senior members of the research establishment now. Thank you very much indeed.
Thank you so much.